calcium that occurs in the cytosol from the endoplasmic reticulum. And we can actually visualize this using some very specialized what we call calcium dyes. So they actually change their color in the presence of different concentrations of calcium. And so when we see this complete massive movement of calcium from the endoplasmic reticulum, we're going to see it be used in a number of particular processes. We have the exocytosis, muscle contraction, they're all calcium-dependent systems, as well as some of the cytoskeletal components within the cell, which with their rearrangement and movement, such as the spectra that we saw inside the inner leaflet of the membrane, causing that partition within local areas of transmembrane proteins, they all require calcium to actually be a constituent in that particular process. Now, we maintain a low concentration of calcium in the cytosol, which is going against the equilibrial constant, because we have very specific pumps which we've been introduced to in regards to the calcium ATP pumps, which specifically move that calcium from the cytosol into an organelle called the endoplasmic reticulum. We also see these types of pumps moving much less calcium, but still moving calcium in the mitochondria and as well in plasma membranes. So there's a dispersion, and depending on what they use and want to use that calcium for, we're going to have different types of pumps which are going to be using either electrochemical energy or a potential uniporter system in regards to moving things in and out. Now, calcium becomes very important when we think about hormonal and neuronal stimuli. So calcium, as we've seen in regards to activation propagation with the presynaptic calcium channels. And the interesting thing is we actually have different types of calcium channels in our terminal butons. We have the N, P, L, T, P, and Q type. And these are classified by their reactivity with other compounds. So we can actually classify them based on what kind of compounds they react with. Some will react with some will react with will not react with others. But their main role is to allow calcium to flux through the membrane. So in those particular components, we have a cellular response, whether it be propagation of an action potential or even activation of other proteins that bind calcium. And we have an illustration here of calmodulin, which is a protein that basically is a calcium-dependent binding enzyme. So if we want to be able to activate certain things, we will need to basically undertake the activation of a calcium, potentially a calcium-dependent enzyme to go through a process of phosphorylation. And the reason this protein is calcium-dependent is because what it does with calcium, it coordinates the calcium moiety in several arms of alpha helices in the structure. And once you get that particular coordination of a number of calcium moieties, it changes the three-dimensional structure of that protein to become then a biologically active conformer. In this case, we like what we saw with the protein kinase A and the cyclic AMP, and how cyclic AMP with those four binding sites impacts the regulation and control of the protein kinase A. We see the same thing occurring in a similar fashion with the regulation of the calmodulin binding, where calmodulin requires four moieties of calcium to bind to its particular structure to give you its stabilized three-dimensional structure and shape. And what we actually see is a kink occurring in regards to the formation of this dimer of the calmodulin structure. So we have, these longitudinal arms, there's two of those, and basically a kinking of those to have two moieties of the calcium bound to then cause it to actually then go around the individual substrate, which is being presented here. Now, calcium also is very important. We have a post-translational modified amino acid that we've talked about in the past, which is called gamma-carboxyglutamic acid. Gamma-carboxyglutamic acid is a gamma carbon at the a carboxylic acid at the gamma carbon of the glutamic acid. And it has then two carboxyl groups that coordinate with another gamma carboxyglutamic acid. So there's 4 carboxyl groups that coordinate calcium. And this is really important in some blood clotting cascade components. But also what we see similar here with the calmodulin, calcium can aid the situation, though it's a non-covalent interaction, through ionic interaction and charge interaction, cause a 3-dimensional structural change in a protein. And we see gamma carboxy containing peptides or proteins forming alpha helices when the calcium is present. When we use a chelating agent such as EDTA, which you might see in your laboratories, that agent sucks up the calcium and binds the calcium. So if you put EDTA either with calmodulin or with gamma carboxyglutamic acid containing proteins which have a 3-dimensional structure, you will actually destroy the 3-dimensional structure because you're sucking off basically the calcium in the system. So again, the chelators have that capacity to draw that calcium away from the structure because that calcium is not covalently interacting, so ionic charge interactions. So again, an illustration of the issue of shape and form and stabilization, in this case with calcium. So we have a whole load of proteins that are regulated by calcium and calmodulin. And again, see some of these are GCPRs and in regards to cyclic AMP, and also some of the ones we're going to see. Let's see which ones are we going to look at. We are going to have a look at some of these. We've already looked a little bit at the sodium-potassium channels, and you can read a little bit in the book about the ones that are in the eye cells in regards to their interactions. We have also these NAD plus kinases. We're going to come to some of these a little later on. The glutamate decarboxylase. So again, these calmodulin-dependent enzymes have a lot of work to do with various biological systems and require that flux of calcium and that actual binding of calcium to give their particular biological activity. You might have heard of this particular protein. So this is another illustration of a protein that we have been utilizing due to its engineering principles in causing fluorescence. So green fluorescent protein, or GFP, is a common element that you'll see inserted into particular proteins. So you will only get kind of a understanding of how these particular proteins are inserted by looking at the issue of the 3-dimensional structure and finding a spot where you can bring in a very large protein. So this is a pretty large protein. I think it's over 130, 140 amino acids in length. And what it actually does, it has a chromophore that has the capacity and the orientation within its beta barrel structure. So we have these anti-parallel beta sheets with some loops, all combining with this particular central chromophore, which provides a situation of fluorescence emission. So we have bioluminescence. You know, you go in the seawater, you shake your hand, you see all this coloration occurring. That's one form of fluorescence. But this is a fluorescence emission transfer. And we'll talk more about this in a second, because it's actually a very useful tool to indicate or even visualize a particular bioengineered protein. So originally it was isolated from jellyfish, and it has a very pronounced presence in molecular biology and biochemical research. The reason being, we can be engineered specifically to produce different colors. So they all retain this natural, beautiful beta barrel configuration with some amino acid changes in the chromophore region, because what we see by engineering it, we can make it go blue, cyan, or red. And this means basically we can stimulate a particular protein that contains this particular fluorescent protein by using a particular wavelength, and it will absorb that wavelength and cause a re-emission of a wavelength at a different frequency, meaning a different color. So in this case, we might be at a particular wavelength outside of 400 nanometers and say this is just the blue fluorescent protein. And basically what we'll see is then a spectral emission. So let's say we're stimulating it at most probably 300 nanometers. we're going to get an emission range between 400 and 500 nanometers with a maximal point about 450. So again, we can stimulate with one particular wavelength and emit a different wavelength in the form of a color. So again, very important, because what we can do with this is use FRET, where basically we have our individual fluorescent proteins and we can stimulate at 433 nanometers, and we see a specific absorbance at 476 nanometers, so a different color. And what we do with fluorescence resonance energy transfer, or FRET, is that we couple this with other overlapping fluorescent proteins. So you can see here we have a number of overlapping proteins: the red, the cyan, the green, and the yellow. Not so much, sorry, the blue, the cyan, the green, and the yellow. Not so much the red. The red is a little bit further out. But because these guys overlap, if they're in close proximity to each other, they're going to basically stimulate the other chromophore. So let's have a look at this specifically when we look at a engineered protein that still retains its biological function, in this case an affinity to two other proteins. So two proteins, one is both are engineered to contain a fluorescent protein part in their primary structure, but they still fold and still exhibit the same affinity to each other. So what we can do, we can utilize our tether in regards to the green fluorescent or the cyan and yellow fluorescent protein. Now, so if this guy is in close proximity to this one and basically binding the two genetically engineered proteins together because they're becoming an associate, we're going to see an interesting transfer of energy to the second fluorescent protein, in this case, the yellow fluorescent protein from the cyan fluorescent protein. So in this case, where normally, if these two are not coming together in close proximity to allow the energy transfer, we're only going to see the absorption or the admission from the absorption. So the absorption at 433 nanometers, we're only going to see an emission at 476. That's if these two proteins are not working together. But if they are working together, and they actually come into very close proximity, we will then cause a stimulation at 433 nanometers, but we will not see a admission at 470 nanometers, like we saw with it being alone. Because what is going to happen, we're going to see an energy donation, because the yellow fluorescent protein spectra, absorption spectra, is in with the range of the CFP, and therefore that we get an energy transfer, and we see a new emission of a new spectra, a new colorization at 527 nanometers, i.e. yellow. So we have this capacity to kind of understand if two proteins are close to each other or not close to each other. And this is commonly used, and you'll see this in the homework, in regards to determining whether the segments on an extracellular transmembrane protein are in the same locality, whether they're on the same side of the transmembrane or of the protein, on the same side of the membrane, on the transmembrane protein, on the cytosolic side. Because what's going to happen, if the other part of the transmembrane protein has the other FP, fluorescent protein, the thing is they're not going to be in close proximity because they're in different orientations. You're only going to get fluorescence if it's on the outside of the cell, because fluorescent light or the UV light is not going to penetrate through the lipids and stimulate the BFPs, GFPs, or whatever on the inside of the cell. It's only on the outside. So if they're not together, in this case, there's no transfer, no signal, and basically no change in the color in regards to the original form. So we use these as actually molecular calipers. And we can do this with our protein kinase A in regards to the reactive clefts and the regulatory sites that we've seen previously. So let's put on a BFP, a blue fluorescent protein, on the regulatory sites, and a green fluorescent protein on the catalytic sites. Now, if these two are in close proximity to each other, we are going to see a stimulation at 380 nanometers and an energy transfer to the GFP at to give us an emission at 545 nanometers. So let's have a look here again. I want to show you this. So we have the GFP and the BFP. Is that correct? Yes. BFP and GFP. And again, so here we have the BFP, fluorescent emission spectra, and then also the GFP, and there's an overlap. So therefore, we can get this resonant transfer occurring in regards to this particular system. So as we know with our protein kinase A, if we start adding 4 moieties of cyclic AMP, we're going to get dissociation of the regulatory site. It's going to bind 4 moieties of cyclic AMP and allow you then to have a soluble component of the catalytic site move away from the protein kinase A to allow it to do its particular job. In this case, when we add the cyclic AMP, we're going to get dissociation. And therefore, what we're going to see is a stimulation at our normal wavelength at 380 nanometers. But we're going to see an emission at 460 because at this stage, we're not in close proximity to cause that transfer when the regulatory site and the enzyme site, the cleavage site, or the catalytic site, were in close proximity to each other. And in this case, we have a clear indication that those two proteins have dissociated and basically are going to be at a distance, not allowing the production of FRET. So with that particular situation, we're not going to see any emission at 545 nanometers. So it gives us a clear indication that these proteins have now moved away, given that they have now had cyclic AMP being brought into their regulatory sites and then causing their dissociation. And as you know, as we remove that cyclic AMP and degrade it, those regulatory sites come back into play, high affinity for the catalytic sites, and we'll bring them back together. And once they bring them back together and we gain shine light at the 380 nanometers, we're going to then see the light emission at 545 nanometers. Now, you're going to have to understand this and apply this to transmembrane proteins. And again, the same situation. If we have transmembrane domains which have the GFP and BFP on the same side, and you put fluorescent light at 380 nanometers and they're in close proximity to each other because they are transmembrane proteins, and they will be, they're going to have then the emission at the 545 nanometers. But if they are in the opposite direction, so we have a serpentine receptor, and this part is the BFP and this part is the GFP, because you can't get fluorescent transfer occurring because of their orientation in the transmembrane protein. So this one's in the extracellular domain, this one's in the cytosolic domain. You're not going to get FRET. So again, a very important tool, which you'll utilize both in your homework and you will see on the exam. Another use of FRET is when we're looking at particularly proteins in regards to interacting with a particular region. Let's say even think of the calmodulin protein we talked about, where we have calcium, which then causes a three-dimensional structural change. We can put the proteins, the fluorescent proteins, on either end of those particular structures. And so when they actually do their folding and come in close proximity, we can also determine whether those things are folded correctly or they're in close proximity to each other. So here we talk about this and we talk about this later on in the phosphoserine binding domains. So in this case, what we have is two sticky ends of the fluorescent proteins, which are not going to cause FRET until they're in close proximity to each other. And in this case, we have 433 nanometers and an emission of 476 nanometers. Now, if we have the activation of the protein kinase A and the serine becomes then phosphorylated, This particular molecule here has a phosphoserine binding site, so once this is phosphorylated, this is going to have an interaction with this. And with that interaction, we're going to move the YFP in the close proximity to the CFP, and then giving us that capacity to produce FRET, where we'll see a change in the fluorescence emission, where we're stimulating at 433 nanometers, and now getting an energy transfer occurring to see an emission at 527. If this did not occur and we did not get phosphorylation and did not get protein movement in the sense of coming together, we would only see the FRET coming off at 476. So again, another illustration of using fluorescence resonance energy transfer, which is a commonly used element. But again, please remember, and this is one of the biggest things that biochemists and biologists fail to think about in some cases. You are inserting a very large protein, a fluorescent protein, into a primary structure. That has its own three-dimensional structure, and it might have consequences on the three-dimensional structure of the target protein. So you've got to be very careful when you're engineering these, because you may lose the biological function of the system altogether. So again, keep in mind, this is an insertion of a new protein into an existing primary sequence, and it has its own characteristics of to retain a beta barrel, 11 beta barrel strands in regards to anti-parallel beta sheets to give you that fluorescence capacity. But that insertion may also influence the primary structure of your target protein. So keep that in mind. So here we have our illustration of how we can use FRET, but in a different system, in regards to the phosphotyrosine binding domains. Now, these are very important in regards to the MAPK cascade, the protein kinase A, protein kinase C, and even a new one called protein kinase G, which we haven't talked about. What they basically do, these phosphotyrosine binding domains, utilize the serine and threonine residues as phosphorylational targets. And through that phosphorylation, they have the capacity then to bind to a particular substrate. So in this case, let's have a look at the phospho-binding capacity of this particular protein, which is basically going to give us the capacity to interact with a phosphorylated or non-phosphorylated constitution. Here we have tyrosines, which are not phosphorylated, okay? But this guy is going to interact with one single phosphorylated tyrosine further away from the active site. So we have a coordinational capacity that's occurring. But within this own protein structure here, we have also another tyrosine site that could be phosphorylated. Now, this is providing you a coordination of two particular proteins that are doing their particular job. But what we have then is a process of autophosphorylation. And with that autophosphorylation occurring, we can get autoinhibition. In this case, what we'll see is the phosphorylational point in this particular protein being phosphorylated and then interacting with itself at the SH2 domain, which is normally reserved for the substrate, and therefore stopping the substrate from binding to the particular soluble protein. Because basically it's tying itself up by that simple event of phosphorylation. Dephosphorylation of that tyrosine will bring it back to its active form and allowing it to interact with the substrate. So that substrate has to be phosphorylated in that particular position. So upon phosphorylation, we get then some interactions with partner proteins, or we triggering a increased mechanism downstream. Now, the interesting thing is with these phosphotyrosine binding domains, they act as regulators, and we're going to see this with a various number of enzymes. This process, again, of phosphorylation, we know acts with regulation in regards to inhibiting and activating, depending on the system. But now we have an insight that autophosphorylation of the active protein or of the active protein can cause its deactivation. And so we have an example which we've come also a little familiar with is the glycogen synthase kinase number 3. So we know in this particular form, we're going to see the glycogen synthase number 3 be active and allow us basically have the glycogen synthase interaction, because it's going to take the phosphorylation of the glycogen synthase at this particular serine position to interact with the enzyme to allow phosphorylation to occur at the neighboring serine positions. This gives us the issue of being able to have an active glycogen synthase. So two proteins coming together, coordinated by a phosphorylation. But with the glycogen synthase kinase number 3, we also have its own serine position close to the active site where we see it interacting with the glycogen synthase. In this case, we can go through a phosphorylation event here, and the tether is just the right length to actually mimic the positioning where the phosphoserine comes in on the glycogen synthase. So it actually acts as a competitive inhibitor. And in this case, we see here where the phosphorylated serine now deactivates the glycogen synthase kinase number 3 through autoinhibition. Again, dephosphorylation of this serine or self dephosphorylation of this serine will allow then this active serine to become hydroxylated and cause then the reactivation of the enzyme. So again, we have these phosphotyrosine binding domains, which become a high level of self and auto regulation. So we're going to have to keep in mind when we look at some of our enzymes and the process of phosphorylation again, in regards to, is it an auto-regulational event? Is it a regulation event due to another protein? Or is it a regulation event due to an inhibitor or an allosteric modulator? So we're now increasing our depth of capacity for regulation, in this case through a simple self-phosphorylation event interacting with the active site to prohibit, importantly, the substrate from coming in to the enzyme system. And you can read more about that on page 468. So we already have this part in mind. So here we have now a clear indication of how we're going to get this guy to be inactive, because the protein kinase B is going to phosphorylate that serine in that glycogen synthase kinase number 3 and cause its own auto-inhibition. So again, in this case, an illustration of how simple phosphorylation has the capacity to then stop the activity of a simple enzyme. So we've already looked through this particular illustration previously, but I'm making the point here where the protein kinase B is playing that particular role to phosphorylate that particular serine to give you then the activity to cause its inhibition. Okay, so it's an illustration, as I said here, of the substrate being prohibited to be bound due to auto-inhibition, and then ideally the dephosphorylation of that serine by another enzyme. And it could be also I think it's the glycogen synthase kinase number 3 that actually can cleave off this serine phosphorylation to then allow it to be the active form and allow you then to reactivate its interactions with the glycogen synthase. So in this case, this is an illustration of enzyme inactivity when phosphorylation occurs, because it's prohibiting the substrate from binding. Okay, so let's have a look a little bit about the lipid membrane rafts and the calveola in regards to signal protein transduction and signal situations. So let's have a look at what happens where we get a change in the lipid membrane, which allows the accumulation of particular transmembrane proteins and peripheral proteins. Obviously, within the sea of lipids that we have this capacity to change the anchoring positions of the proteins by giving them an environment which is more suitable so they can actually physically cluster. So in these lipid membranes or lipid rafts, they're rich in sphingolipids and sterols, which give us this capacity to change both the thickness and the permeability in regards to the stabilization of proteins within those two leaflets. So some proteins are highly localized in these particular rafts, and we have some epidermal growth factors and some other transmembrane proteins. And we believe, and this is still kind of open to controversy, is that they play a regulatory role. So let's say you have an enzyme system where you want to move things across a number of enzymes very quickly. It is no good having these systems at very distant regions, because you've got this issue of diffusion, because those molecules or those substrates that one enzyme system is producing that wants to get to this enzyme system can diffuse across the system and be diluted down. So it's actually best to bring them together in very close proximity, so you can use FRET to measure this, and move the compounds between the two enzymes to do the work more efficiently. So we believe that there's a regulatory role in regards to bringing enzymes together to do their work. And we can actually cause the distributional changes of these lipid rafts and these local clusters of enzymes in the membrane by just simply changing the cholesterol concentrations in regards to their change, and also sphingolipid concentrations in regards to disrupting the normal signaling pathway. So, as I said, it increases the probability of interactions, particularly of those molecules that are being produced within the same localized raft, which are being consumed by another enzyme in that particular raft. Some rafts also act as membrane transporters, particularly in the epidermal growth factors. So we have these lipid rafts that are basically going to be utilized as transports to congregate those types of proteins and basically mop them up. And because they are more suitable to be like with like and be embedded in the lipid raft, they're going to congregate and have a larger mass of those functional proteins together. We've already talked a little bit about the caveoli in regards to the dimerization to allow you to have the sphingolipid layers and a different type of fatty acid in the outer leaflet of the membrane to give you this puckering component with the caveoli forming the dimer. With that, we have a capacity. So out here, let's say it's the extracellular domain, it's a very dilute environment where if we're producing a molecule that needs now to go to an enzyme, it's going to be basically a bit of a probability hit and miss to get that little compound all the way to the enzyme where it needs to be. So with this type of system here, they might have an enzyme transmembrane protein system in a lipid raft here, and an enzyme protein system here with a lipid raft here, where this particular compound taking the individual product to produce a substrate, which then becomes the product, which then takes the substrate to become the product. The product then becomes the next substrate for the next enzyme. And instead of going all the way through the transmembrane domain and trying to find that, it's kept very close proximity to each other. So there might be a microcosm of concentrational differences of certain substrates within these caveoli because they're being swapped between different enzyme systems that are being present within the caveoli due to the lipid rafts, but also due to that puckering to allow a high density concentration of these molecules instead of the lesser density concentration of molecules that would happen in the open environment of the ectoplasmic, endoplasmic, exoplasmic, endocellular plasmic domain. Now, the caveoli also respond to phosphorylation and insulin of the tyrosine moieties within the dimer. And this is basically what causes then the puckering component to occur. So we have these capacities now, as we saw with the insulin receptor being phosphorylated, integrating the insulin substrate number one, doing its thing either by increasing the transporters through the protein kinase B, exacerbating the GERB2 receptor to increase the MAP cascade effect to basically promote the extracellular kinases to move into the nucleus to increase then protein transcription to allow protein expression to be increased. But again, we can utilize these lipid rafts to give us a more sensitive impact, given we can concentrate those receptors. Because another thing is with lipid rafts, because we have the concentration of those particular receptors, when we have a very small extracellular stimulant, it's going to have a better probability, instead of just finding one receptor, which is very distant and very sparse. If it hits a particular layer where, or a region where there's very numerous numbers of these same receptors, which is looking for that particular ligand, it's going to be able to increase the capacity and probability of that ligand making a stimulation in a transductional situation with a GCPR or a receptor or an ion channel. So again, several overall lipid rafts enhance signal protein interactions and make signaling a lot more efficient by crowding things together, by providing them, I'm going to put it in the nicest way, the right soil type to grow, meaning that the lipid raft provides an environment which allows transmembrane proteins to conglomerate together to be more efficient in their particular job and their work. Okay, so the better soil type, the better growth you'll get, basically. So here we have also some illustrations in regards to signal components that are present in various phyla. And as I kind of told you before, when we look at the KCSA protein, which is a potassium channel, we see potassium channels across mammals, plants, and bacteria. And so this is where we can take a toxin that might kill a mammal, maybe a scorpion toxin, and can interact with a bacterial ion channel because we have similar three-dimensional domains and binding efficacies and interactions of the individual proteins or peptides to these receptors or ion channels. But as you can start seeing through the whole list, there's some evolutionary aspects that are highly conserved, but there are also ones we are not sure about. And this is the way science is. We don't know absolutely everything, though we might be screening bacteria for, in this case, adenyl cyclases, but we haven't screened all bacteria for adenyl cyclases. So there might be one that might actually have adenyl cyclase. So we can't say for certainty that it's not present. So we have a whole load of these particular ones, and we've come through some of these already, and we're going to look at the two-compartment His-tag kinases in a second. But ultimately what we see our calmodulin, the MAPK cascade, we see our IP3 ion-gated systems, which I haven't really talked about, where IP3, if you think about an ion gate system, which has a cytosolic ion gate for calcium, by again increasing that calcium load, you might also change that ion voltage sensing gate because you're changing the polarity in the cytosolic domain by changing the amount of positive ions being present. Because you've got these little endoplasmic reticulums with all these positive calcium molecules, you're flooding them into a negative environment and changing then the membrane potential. So again, you have the capacity to basically utilize the calcium to influence the inner membrane potential, which will then also affect the membrane potential across the normal cellular membrane. So there's a whole lot of things that we need to kind of keep in mind of what's going to occur when we start looking at some of these transmembrane proteins, their function, how they integrate with the lipid membrane, and ultimately what they do when particularly certain elements such as calcium and other stimulatory components such as peptide hormones, steroids, sterols in regards to their conjugation to produce these lipid rafts to maximize their capacity to provide you the signal elements. So here we have an illustration now of a compartmentalizational system where basically two components are required to get a particular stimulation. And this is usually a phosphorylational event. So the first off, we'll see E. coli with these little flagellal motors that basically keep the E. coli moving around. Now, people originally thought it was just a random process, but actually it's not. It's actually a very attuned system in regards to its biological sensing and biochemistry within inside the cell. Because what it does, it provides you a chemotaxis or a chemical sensory component in the E. coli itself as a bacteria. So E. coli respond to nutritional gradients because basically they want to follow where the concentration of the most food is so they can consume it and grow. But they need to be able to give information to the flagellal motor to say which way do you need to spiral, whether you need to put me in this direction or do you need to put me in that direction. And so what they do, they have a histidine dependent phosphorylase, which is a transmembrane protein, usually at the head of the E. coli. And basically, they are determining whether a phosphorylation event should occur or not by the stimulation of an attractant. In this case, once we get the attractant, the A, binding to the histidine transmembrane protein, that histidine will consume or the enzyme cytosolically will consume ATP and transfer that phosphorylation group from ATP, producing ADP, and phosphorylate that histidine. The histidine. Now, the phosphorylation of that histidine then causes a new activation, where basically that histidine phosphorylation is going to then take another protein in regards to a regulatory protein and allow that to actually transfer that phosphorylation group onto the aspartic acid. In this case, this soluble aspartic acid then has the capacity to influence the rotoring, the rotor motor, and we'll have a look at this motor later on, in regards to its phosphorylation event, and change then the function of the movement of the flagellal motor. Now, in the same process, we then cause dephosphorylation of the transmembrane histidine attractant. We're going to get this basically diffusing off because no longer is the concentration gradient, and then it being recycled to become the active component, as we see in the front of the head. Because remember, these guys have the capacity to move along the membrane. So here we have this sensing component using phosphorylation and then transferring that phosphorylation into a soluble protein that is going to then be phosphorylated, and that phosphorylated soluble protein then interacts with the flagella to give it a particular action. So we have the attractants that bind to these histidine kinases. We phosphorylate those histidine kinases. We transfer that phosphate group cytosolically to the aspartic kinases, and that aspartic kinase basically then is a responding, regulating protein through its phosphorylation event. Again, a three-dimensional structural change caused due to that phosphorylation event, and then that will actually then have a high affinity and bind to the cytosolic side of the flagellal protein and basically make the flagellal component change a movement or stimulus in regards to its activation. So if directly head on, they will continue. So with a continual concentration is at the head, and we get a continual stimulation with the phosphorylational aspartic acid, we're going to move into a particular straight line. But if the head now starts to say there's a lesser concentration or no concentration, it's going to go through the process of not having this aspartic acid phosphorylated and therefore changing the direction of the flagellal motor. So it changes direction, and then once we get to the right head direction of the concentration, we're going to get the re-stimulation to go us into the straight direction. So if we don't see this stimulation at the head of the E. coli, we get a tumbling effect because of the dephosphorylation event here, no longer stimulation of the flagella to put it in the straight direction. So we get a new direction. Basically, we're reassessing the position based on the concentration gradient of what has been presented with the histidine phosphorylational kinase. So we get this tumbling until that head position starts getting to the right concentration, chemotaxic concentration of food, and therefore we then move into that straight direction. So if you ever see there's some videos, if you go look at a YouTube video, there's some videos in regards to E. coli movement. You can see this tumbling and movement, and then moving around, tumbling, moving around, tumbling, moving around, tumbling. Now, it's not a random path, but it's biased towards that concentration gradient. And so again, by using two enzyme systems, we can now influence a motor protein, which this is basically a motor protein, and influence how it moves, and then how it moves the whole body of the E. coli. So it's a very interesting illustration of a compartmentalization system using enzymes, using a initiation to determine a concentration gradient stimulus outside the cell to influence a motor protein from the cytosol side, which then influences the movement of that particular bacteria. We also have this occurring in plants. Now, we eat plants. Let's say we will take the apple. So the apple basically is a seed carrier with enough nutrients to support that seed if it's lucky enough to go through the right environment to get germination to occur and bedding in the soil. So if that does not occur, what happens, the seed or the fruit rot and basically break down the materials. But that rotting is actually a good thing for the seed because it's providing carbohydrates, promoting the process of germination. But what we do, we prohibit that process by taking the apple when it's nice and mature and eating it. And again, eating those carbohydrates for our own dietary components. But what they do, plants actually utilize ethylene gas via two-compartment system using a MAPK cascade to increase protein secretion to exacerbate the breakdown of those celluloses and starches to increase carbohydrates for the cells and for the seeds to basically start their germinational process. So we understood this very early in regards to fruit ripening. So if you think the old saying that one apple spoils the barrel, it actually does, because that spoiling apple actually produces ethylene gas to basically cause then the other fruit around it to actually ripen and go bad. But what we try to do is inhibit this issue in regards to promoting or removing ethylene gas from those particular fruits. So commercially, we pump ethylene gas in holding rooms before shipping particular fruits like bananas to market. And what this does is basically prohibits or increases the protein expression, increases protein starches and carbohydrates. So what we have with the absence of ethylene gas, and you've seen those green plastic bags you can put your fruit in, what they basically do is basically prohibit the transcription of those enzymes that would be normally breaking down the starches to give you that rotting process. So by having the absence of ethylene, we are basically activating a protein kinase which inhibits the MAPK cascade. Therefore, we get no transcription occurring. But with the presence of being able to ripen your fruit very carefully, we activate that particular cascade and increase transcription to ripen the fruit. So if you think about your bananas you get from Safeway, so your bananas are usually very, very green, and you taste them green, they are floury, right? They're kind of starchy. Exactly what I'm talking about, starches. But as they mature, they get those little brown spots very quickly, because what they've done in those, when you see someone take those bananas out, they're in these special plastic bags, and those special plastic bags are basically trying to basically get that ethylene gas in regards to basically providing you the inability to produce ethylene response proteins. So here's our illustration in regards to our ethylene receptor. We have ethylene gas binding to the ethylene receptor, which is then going to inhibit the CTR MAP cascade approach, which will then inhibit the process of ethylene proteins, which are usually going to be enzymes that are going to promote starch breakdown and increase carbohydrates. So in some cases, you might see localized ripening, and you can do this with your avocados. So if you take an avocado and you put it in rice, so put your avocado, which is not ripe, but put it in a half container of rice, so half of it sticking out and half of it sticking under the rice, because the rice is basically acting as a barrier, so the small amount of ethylene gas that is being produced is being actually moved away it's being contained. You're going to promote the process of non-degradation, where the ethylene gas being moved away is going to actually promote the process of ripening. So we also see a two signaling compartmentalizational system occurring also in immune responses. I don't want to get into too much of this, but plants do have an immune response, particularly to bacteria. And in this case, what we see is basically certain proteins being expressed by the invading bacteria, which are very sensitive to transmembrane proteins in the sense of these bacterial phage and bacterial flagella sensors. And they had to form a dimerization to basically interact with this case, maybe a flagellal protein number 22, to actually work and promote a phosphorylation through a transmembrane protein. This again affecting the MAP cascade, increasing transcription, but instead of increasing transcription for carbohydrate production enzymes to break down carbohydrates and to metabolize carbohydrates, it might also be secreting systems which will actually increase the concentration of proteins which are responsible for the immune response in plants, particularly our study plant, which is called Arabidopsis. We see a similar situation in regards to mammals with the lipopolysaccharides and also flagellal proteins of this, such as E. coli, and again, a more complex situation occurring in regards to allowing these to interact, form a dimer, go through various phosphorylation events, really focusing on the MAPK cascade to increase certain expressions of proteins which are basically involved in the immune response. That's what I want to kind of say about the issue with the signaling trans pathways and two compartmentalization systems. We have the ethylene gas, we have the E. coli, and then we have these systems in regards to plants in their immune response, in particularly interacting with a cascade, which is going to be pretty promiscuous in regards to producing a lot of different factors and a lot of different proteins, not just those that are affected in the issue of carbohydrate degradation. So we looked at some interesting terminology in regards to desensitization, the beta-arrestin, the secondary messenger system, how we see AMP and cyclic AMP, serpentine receptors, G-coupled proteins, calmodulin, fluorescent green proteins, fluorescent resonant energy transfer, or known as FRET, glycogen synthase in regards to auto-regulation and auto-inhibition, and the two compartment signaling system, particularly focusing on chemotaxis in E. coli. But I really want you to understand how we regulate the beta adreneric receptor and how we desensitize it, moving away from the receptor, from the surface of the membrane. I want to see also your understanding about the central role of AMP as a secondary messenger. You need to understand and draw it. Okay, so I'm putting some specifics down here. You need to be able to understand the integration of the GCPR system as well as the IP3 system, particularly with hormones, peptide hormones or peptide transmitting substances that have the capacity to interface with the protein kinase C by the stimulation of the IP3 with the phospholipase C system that is embedded in the lipid membrane. I also want you to understand how we can measure these types of interactions of associated proteins and their localities in regards to FRET, and also how FRET, if we're using the specific example of the protein kinase A, how FRET will be interfaced and modulated by amounts of cyclic AMP. We also talked about phosphorylation in regards to now being an auto inhibitor, where enzymes that would use a phosphorylation situation to interact with a protein substrate may actually also phosphorylate their own part of their protein and allow that to be actually then become a process of inhibition through competitive binding. But also they might result on an external phosphorylation event, like we see with the protein, with the glycogen synthase kinase number 3, and see its inactivation in regards to the process of autoinhibition. We talk about the segregation and signaling proteins, which we think in regards to E. coli, and the two compartmental systems in regards to ethylene riping and bacterial chemotaxis. So a few things to think about in regards to studying for the homework and for the exam. So let's now get on to a now transitional stage